How does CRISPR work, and why is it important for human health? Today, we're going to talk about CRISPR. CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeats. Before we talk about CRISPR, we need to discuss DNA, RNA, and what is called the central dogma of molecular biology. Our genetic code is encoded in DNA, a sequence of nucleotides with the letters C, G, T, and A. C stands for cytosine, G stands for guanine, T stands for thymine, and A stands for adenine. Our DNA has two strands, which are called complementary to each other. This means that C on one strand matches with G on the other strand, and vice versa. In addition, A on one strand matches with T on the other strand, and vice versa. Our genome is made up of 3 billion pairs of these four bases. The central dogma states that DNA can encode a complementary strand of mRNA, and this is called transcription. The mRNA can then leave the nucleus and enter the cytoplasm, where it can be produced by encoding for chains of amino acids. This is called translation. Now that we've had a crash course on the central dogma, let's go back to CRISPR. CRISPR was originally found to be a defense mechanism that bacteria use to protect itself against viruses. Viruses attach to the bacteria and inject their DNA into the bacteria. The DNA merges with the bacteria's original DNA and provides a type of memory against past viral infections. This occurs in two phases. In phase 1, or the acquisition phase, a virus attaches to the bacteria and injects DNA into the host cell. The DNA is incorporated into the CRISPR locus between a series of special repeating sequences. This DNA sequence is then transcribed into a matching transcript called messenger RNA. This messenger RNA joins with a special guide molecule called trace RNA and the Cas9 protein, and they break off into individual subunits inside the bacterium. In phase 2, when the bacteria is infected with the same type of virus, the Cas9 protein and CRISPR RNA complex recognizes the viral DNA because it is an exact match or complement to the CRISPR RNA. After it is bound, Cas9 causes a breakage of the DNA, effectively getting rid of the viral invader. How does Cas9 perform its function of cleaving DNA in the precise location necessary? Short sequences called PAMs, which are protospacer adjacent motifs, are important in this process. The PAM is a sequence of 2 to 6 base pairs that immediately follows the DNA sequence that is to be targeted by Cas9, as shown here. Here, you can see that the sequence to be targeted is directly adjacent to the PAM. The target location for cleavage of the DNA is identified by an approximate 20 nucleotide sequence that is adjacent to the PAM site. Cas9 will then unwind the DNA strand with the assistance of PAM. This unwound portion can then be cleaved, causing what is called double-strand break. This means that both strands of DNA are cut at around the same base pair location. Cas9 ensures that the correct sequence is cleaved because the nucleotide target is identified by the PAM. After the DNA strands are cut, Cas9 can glue the ends of the DNA together through the process known as non-homologous end joining. This results in the removal of a specific segment of the DNA. Alternatively, scientists can insert new genes into the DNA by using a repair template in the process called homology-directed repair. Here, a segment of the repair template that travels with the CRISPR Cas9 is transferred to one of the two DNA strands. Then, a matching template will be produced and transferred to the other DNA strand across from the repair template to repair the DNA. A researcher can even better target a specific site in the DNA for gene editing. One way of doing this is to use a plasmid, which is typically a circular piece of double-stranded DNA. What scientists can do is insert the Cas9 DNA into a plasmid and then inject it into a cell where they want to edit the genome. The DNA is transcribed into mRNA and then translated into a protein, and this is the Cas9 protein. Cas9 also needs to be guided to the correct place on the genome to make its edits. Therefore, scientists also inject what is called an sgRNA, or single guide RNA. This sgRNA contains the tracer RNA and the crRNA. As you remember from earlier, the crRNA, or CRISPR RNA, is a 20 nucleotide sequence that is complementary to the target DNA, or the DNA segment that we want to edit out. Also remember that the tracer RNA, like before, is a scaffold that binds the DNA and CRISPR RNA to Cas9. Together, the guide RNA and Cas9 can edit out or excise a defective segment of DNA that might be a cause of a genetic disease. What kinds of diseases might CRISPR be useful to treat? These are genetic diseases caused by a mutation in the genome. These mutations are often passed from parent to offspring. One such disease is Huntington's disease. It is a progressive brain disorder that causes uncontrolled twitching movements of the body, leading to an inability to walk, swallow, or speak. People who have Huntington's disease start to develop symptoms in their 40s and can die in their 60s. Huntington's disease has an autosomal dominant transmission, which means that the offspring have a 50% chance of getting the gene, and a single copy of the mutated gene is enough to cause the disease. People with Huntington's disease have a mutation in the HTT gene. This mutation is called the CAG trinucleotide repeat, which appears several times in a row. This means that there are many repetitions of the nucleotides cytosine, adenine, and guanine. In Huntington's, you can have 40 or more CAG repeats in a row. This abnormally long segment in the HTT gene causes small fragments of protein to accumulate, bind together, and accumulate in neurons. This is toxic to nerve cells and disrupts neural function, and ultimately leads to death of the neuron. CRISPR was used in a mouse model of Huntington's disease to edit out the mutated HTT gene segment. As you can see from this fluorescence image, the green fluorescence correlates with the toxic accumulation of Huntingtin protein in the mouse brain with Huntington's disease. After using CRISPR, the green fluorescence is much reduced, indicating that the mutated part of the Huntingtin gene was excised. Human trials of CRISPR to treat Huntington's disease are currently underway. CRISPR has great potential to treat genetic diseases. In fact, the potential of CRISPR was recognized by the Nobel Committee, who awarded the 2020 Nobel Prize in Chemistry to Jennifer Doudna and Emmanuel Charpentier, who were the discoverers of CRISPR. Now you know what CRISPR is, and the next time you hear about CRISPR, it might be in the context of new treatments for genetic diseases for humans.